Hello everybody. In this first part of this video, I'm going to introduce you to the methodology that is known as a line transect. And this will also be an introduction to how you will be collecting the data for your next report. Here we go. So you have already read about a line transect in your book or in the handout that covers the option, but a line transect is a method that you can use if you want to analyze the effect that some kind of abiotic factor or perhaps multiple factors might have or could have on the plant community. So what you'll do is that you will draw a line, not draw, but you would decide upon a line, upon a transect, as it's called, and this should span across an area where you think that there is a difference in at least one abiotic factor. So in this example, we would expect that there is more light out here in the grassland than inside the forest, for instance. So we place our line transect along this line here, and then we place quadrats along the line. You would of course just have one quadrant carrying one quadrant around, and then you put it down out here and figure out which plant species are found here, and then you move a little bit, figure out which plant species are found here, move again, and so forth. So this is known as a line transect. And the abiotic factors that you can measure, because we don't really have the equipment available that we would normally have if it was a school field trip, then we would be able to measure the pH and lots of different things. But what I have decided that should be possible for you to do is that you could download a Luxmeter app to your phone. There are many of those out there, and many of them are just free. And you can use that to measure light intensity. You can also dig a small hole in the soil and then assess the color of the soil, because we can use the color of the soil to say something about how much organic matter is in the soil. Because if it's very dark, almost black, then there's a lot of organic matter, and if it's more brown, it means that there's less organic matter in the soil. And we could also assess the texture of the soil by looking at how moldable the soil is, and this would indicate something about the grain size. So if there is a lot of sand would be larger grains. If there is a lot of sand in the soil, it would not be moldable. And if the soil consists of smaller particles such as clay particles, then it will be much more moldable. So you can assess that, and then you could also do some kind of assessment of your humidity of the soil. Is it a very moist, very humid soil, or is it a very dry soil? So these are the abiotic factors that you will be measuring. And why is grain size and humidity and the color of the soil, why is that important, or how can we use that to say anything about the plant community? Well, I have tried to produce a picture here that hopefully can explain this. So I'm going to try to give you a little bit of soil science within 30 seconds here. And you should know that soil is very complicated. You can spend years studying soil and soil development in the university. So this is just a very, we're just really just scratching the surface here, right? But what I've tried to do here is this triangle, right? So and in the center here, we see that in here we have the rate of breakdown of organic matter. And organic matter, all the dead leaves, all the dead insects, whatever, all that organic matter is broken down by saprotrophs and detritivores. And especially all the saprotrophs, all the bacteria in the soil, are very much affected by both the humidity of the soil and the pH of the soil and the oxygen availability in the soil. And if there is not that much oxygen, and if the pH is acidic, and if the soil is very humid, then the rate of breakdown of organic matter will be very slow. But if the soil is more dry and the texture is more sandy and the pH is neutral, then a lot of organic matter will be broken down quickly. Yes. And why is breakdown of organic matter important? Well, it is because if the organic matter is not broken down, if it stays there, then the inorganic nutrients that the plants need to grow is not available to them because it's just still trapped in those organic molecules. And if the rate of breakdown is fast, then there is more nutrients available to the plants growing in that area. So the texture of the soil tells us, of course, something about the oxygen availability. If there's a lot of very small soil particles and if they're very densely packed together, then oxygen cannot sort of move into the soil. There are no air spaces in the soil. And if the soil is like that, then maybe rain cannot drain through the soil, so maybe the soil would also be very wet. So the texture of the soil can tell us something about both the oxygen availability and could also maybe say something about the humidity of the soil. The color of the soil we can use to directly sort of assess how much organic matter is in the soil, because if the soil is very black or very dark brown, this indicates that this shows that there is a lot of organic matter, because the black color is the only partly decomposed organic matter. And if there is a lot of only partly decomposed organic matter, then that would most often lead to a low pH, which would again have a negative effect on the rate of breakdown of organic matter. Yes. Was that 30 seconds? I don't know, but we'll move on. So just to give you some examples of what your line transects could look like. So this is a meadow, and down here we have a little stream. So you could have a line going from the little stream and up the hill, for instance. That would be one example. And the abiotic factors that would vary along a line like this would be humidity. So we would expect high humidity down here, lower humidity up here. We would then again, due to the humidity and low oxygen availability, because the soil down here might be waterlogged, we would expect a low rate of breakdown of organic matter down here. So we would expect black soil with a lot of organic matter down here and more brown gray soil up here. And it says humidity again. That's my mistake. Well, yeah. Another example is this beach. So you could draw a line that starts at the shoreline and then moves into the vegetation. And in this case, we would see salinity would be an abiotic factor, right? So we would expect the salinity of the soil to increase when we move closer to the ocean. We would also expect light intensity and also temperature to increase, at least down here at sort of the bottom of the vegetation. As we move out here where there is no shrubs and no trees and stuff. And then on the other hand, we would expect species to compete more if they grow in here. Yes, and out here we would expect only few species that are adapted to this very special environment that we have out here. Yeah, and again, just to show you, so what you would do is that you put down, you quarter it at regular intervals. So this could be 2 meters in between, or 2 big steps in between, or something like that. Yes, and here you see my transect. And later on in this video, I'll take you there to that area. But for now, I'm just going to show you that the transect runs from I actually have my first quartered up here on the top of the hill, and then I move downhill to investigate the change in vegetation and the change in those abiotic factors. So what I did before I went there was that I produced a quadrant. This is approximately 50 times 50 centimeters because I need a bigger quadrant for this type of analysis simply because the area is bigger. And especially if you also want to investigate the small shrubs and perhaps trees and stuff, then you would of course need a bigger quadrant. You can't produce a quadrant big enough for a tree, but I think you get the idea. And what I used to do this was simply 4 sticks and then some duct tape. Then of course you also need to figure out where you want to go. And like I said before, it should be an area where you expect abiotic factors to vary along the transect. And in this case, I expect this area down here to be pretty humid, and then it would be pretty dry up here. So I expect that definitely humidity would vary along this line. Then you should also bring pen and paper and your phone and a small shovel so that you could dig a little hole to have a look at the color of the soil and the texture of the soil. Then you should download a lux meter, like I mentioned before. And then I would also recommend that you download an app called iNaturalist. Come on. Here we go. Yeah, so this is just one example of what a lux meter app might look like. And this is the iNaturalist app. And what is neat about this is that you can take a picture of a plant or an insect for that matter, and you can then search for possible, or you can search for possible names of this species. And this app is actually pretty good. It was able to recognize quite a lot of the species that I found on my field trip. So it comes up with possible suggestions, and you could then choose the one that you think fits the best. So I very much encourage you to use the app, and if not, then you could maybe borrow a book to look up some of the plants, or you could also simply name them species 1, species 2, species 3, and so forth. And then of course remember to take pictures of all the different species that you find. Yes, that was the introduction. So hey there, and welcome to this introduction to how to perform a transect analysis. I have moved myself to Ørkilsborgen, which is an area that contains the remains of a very old castle near Svendborg, and I will be performing my transect analysis here in this area. I have brought with me a homemade quadrant. This time it's quite a lot bigger than it was the last time. I've just used 4 sticks and some duct tape to produce it. And I haven't measured the exact area yet, but I'll do that when I get home. My estimate is that it's maybe 50 times 50 centimeters this time. So it's bigger because the area that I'm covering is bigger this time. I also have with me a small shovel, and then I have of course pen and paper to collect my results. And this time what I need is this page. So I'm gonna explain my transect to you in a minute, and my transect will be divided into 10 zones. So there's zone 1 and 2 and 3 and so forth, and in each of those zones I'll be putting down my quadrant and recording the plant species that I find. And this time we're not going to count, at least not here in the beginning, we're not going to count how many of each species, but I'm just going to record the names of all the species present in my quadrant. So that's one part of the analysis. In each of my zones I will also be investigating some abiotic factors, and the factors that I have decided that you can measure without all the equipment that we would normally bring on a field trip are these. So if you download a lux meter app to your phone, you can actually measure light intensity, so we'll try to do that. Then you can assess the color of the soil, the texture of the soil, and the humidity of the soil, and I'll explain to you a little bit on how to do this. And there's also some pictures that you can use for doing this in the report handout that I'm going to upload to Lecture. So that's basically the materials that we'll need to collect the data. So my transect is this. I'm going to move from the top of the hill to the bottom of the hill. So what I do is that I sort of draw a mental line from where I'm standing towards the little door down there, or towards the pond. So that's going to be my line. Normally we would maybe stretch out a piece of measuring tape or something like that to sort of make sure that we follow that line. But since we're going to try to do this without access to lots of materials, this is what we'll do. So we'll draw a mental line from one easily recognizable spot like the top of the hill to, for instance, like this, the bottom of the hill. So this is my transect, and I'm then going to try to divide it into 10 zones of sort of equal length. So my zone 1 is where I had placed my quadrant, and then I'm going to take 2 long steps, and then I'm going to place down the quadrant again in zone 2, and then two more steps downhill, and that would be zone 3, and so forth. So that's how I'm going to do it. But as I said, I have already placed my quadrant in zone 1. So what I'm going to do is that I'm then going to take a close look to see if I can identify the different species that I have in my quadrant. And I have at least one species of grass, and it seems like there is another species of grass over here, so I can refer to this as grass 1 and this as grass number 2. And then I have a little, I don't really know the English name, I can look that up when I get home. This is in Danish called storkenape. So that could be my species 3, and I would then of course make sure that I take pictures of all the different species that I see. So I have at least these 3 species. Do I have more? I'm gonna take, then I have a trifolium, in Danish kløver, over here. So that's also in quadrant number 1. And that seems to be it. So for plants present in quadrant number 1 or in zone number 1, I would note down grass 1, grass 2, species number 3, which is storkenape, and then some trifolium, the one I had over here. And then to assess the abiotic factors, I'm going to dig a little hole, and I'm pretty sure I'm not allowed to dig in this side, so I have dug a very, very small hole. I'm going to cover it all up again, but I'm going to take a look at the color of the soil, and this is brown gray. It looks a little sandy to me. And the reason that you need to dig a little bit is because we want to assess soil quality where the in the depth where the plants have their roots. So ideally 15 centimeters down, but if that's not possible, then just dig a smaller hole like I've just done here. So I'm gonna write down that this is gray brown and that I can see grains of sand in it. So that would be some observations, and then I'm gonna try to see if I can squeeze it together to form something. No. So the texture is, I would say, not moldable for this one. And then I would say that even though it's a rainy day, when I dug down 5 centimeters, the soil was actually pretty dry, so not very humid up here. And for the light intensity, I'm going to use the app, the Lux meter that I have downloaded, and I'm going to measure the light intensity at ground level, so where the plants are growing. And this will of course vary quite a lot. So today it's really a cloudy day, and if it was a sunny day, of course the light intensity would be higher, but it would still give us some impression of whether the light intensity varies down the transect. So I'm now in zone 4, and I think that you can see it just by looking at these pictures already, that there seems to be a higher number of different species in this area. So I have at least one type of grass, and then I have another type of grass down here, and then this is a carex. Not, or is it? No. Yes, it is. It's a carex. So this is actually not a grass. And then a third type of grass over here, and some different herbs down here. And again, I would of course take pictures of all the different leaves And I would also encourage you to download and use the app called iNaturalist, because that's actually pretty good at identifying the different species. Then you just take a picture of them and ask the app to come up with suggestions, and then you can use the top suggestion to name the different species. And if that is not possible, then again you refer to them as species 1, species 2, species 3, and so forth. And then of course if you find the species that you have already seen before, then it's still, if I find the same type of grass as I did in quadrant number 1, then that would still be grass 1 or grass 2 or the little stalkernape that I found earlier, or whatever, right? So it's important that the name of course sticks with the species that you're investigating. So in this quadrant number 4, in zone 4, I was actually able to identify 11 different species when I had a close look at it. So I'm now in zone 9, and I have placed my quadrant once again, and if we take a look at here, we can clearly see that it seems that humidity is one of the factors that is probably is varying from the top of the hill to the bottom of this hill. So it's much more humid down here, and I'm pretty sure that if I dug my hole here, the soil would be much darker, probably almost black down here, and also more moldable, I would expect. But since I'm not really allowed to dig here, I'm not gonna dig another hole. I've already dug one at the top of the hill, so I'm gonna stick with that. But if we take a close look at the species here, then we have another carex, a bigger one than the one we had before. But it actually seems that there are not that many different species. It seems to be a huge snail crawling around down there. But it actually seems to be the only species in this area. So I'm going to name this species Carex 1, and then there's just this one species in zone 9. So guys, since you are already moving outside, bringing your quadrant and all that, I have decided that besides collecting data for the line transect analysis, you will also be collecting data that you can use to calculate a Simpson diversity index, because we will talk much more about biodiversity later on, but you might as well collect the data now that you're out there. So you will also be asked to calculate the Simpson diversity index for 2 of your different zones, 2 of your quadrants along your transect, and that would also be part of the report. Yeah, so like I said, we will talk more about biodiversity later on. So what you should know for now is that the Simpson diversity index is a number that we can calculate that tells us the level, so to speak, of biodiversity in a certain habitat. And there is a formula up here. This is just a picture from your book. So there's a formula up here. So the data that is needed to calculate the index is the D over here, is the total number of organisms in the habitat. That's the big N. And then the number of each species, the number of individuals of each species in the habitat as well, or in that quadrant. So, and why would we want to calculate something like that? Well, that's because sometimes biodiversity is not that easy to just directly assess. And this is a picture from BioNinja showing 2 different plant communities. And you can see that in those 2 communities, we have the same number of species. So we have 4 different species in community number 1, and we have 4 different species over here in community number 2. So which one has the highest biodiversity? Well, they have the same number of species. Well, but that's where the concept of evenness comes in. So the species richness tells us something about the number of species in each habitat, but the evenness tells us about the distribution or the frequency of the different species. And if we take that into account, then we would end up figuring out that this actually has a higher level of biodiversity over here, because the species are more evenly distributed, compared to over here, where this species here is a very dominant species, perhaps eventually outcompeting some of the others. But even if it wasn't outcompeting the others, because there is a more dominant species over here, this would be considered less diverse than the other one. So there's a little video on how to collect data that you can use to calculate the Simpson diversity index as well. So I'm still standing on the hill in Svendborg, and I'm done performing my transect analysis. So I'm now going to move on and calculate the Simpson diversity index for 2 of the different zones in my transect. So I have moved to zone number 5 and placed my quadrant in zone number 5. And the data that is needed to calculate the Simpson diversity index is a list of all the species present in that quadrant and the number of those species. So we will try to figure out the number of individuals of each species in this quadrant. That's not always an easy task, but we're going to try. So I'm moving closer to my quadrant, and I have a little purple one that's in Danish called ærenpris. So I'm going to note that down. Then I have a bit of a ranunkel. Then I have, what do I have? The remains of the dandelion. That's the ranunkel again, ranunkel, and then this is called viper. And what else do we have? That's leaves from the viper, and that's the ranunkel. Then we have one type of grass. I'm gonna name that grass 1. And we have this taller grass. I'm gonna name that grass number 2. And do we have anything else? No, I think that's it. Then I'm going to try to identify how many individuals of each. So for the dandelion there was just 1. For the ærenpris there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, this one. And then I'm going to continue to do that. And of course for the grasses it's pretty difficult. But if they stand like this, then you can assume that all these leaves belong to the same individual. And then perhaps for this one, so again it's going to be a rough estimate, but you do it as good as you can. So a list of all the names of all the species and the numbers. And when I'm done with the first quadrant, I'm going to move to the top of the hill and put down my quadrant again and do the same thing.